Thanks very much. And first of all, thanks to the organizers for placing me after Alon in this session. It always makes my job so much easier. So it's much appreciated. So today I'm going to be talking about our reprogramming data set and how we are trying to integrate network inference with that lineage tracing to understand more about these state-fate relationships that I'll get into. But in the middle of the talk, I'm going to take a detour into some traditional developmental biology, just to give you a bit of a background for how we've been designing this platform. And so to back up a second, reprogramming cell identity. So we know that cells can be reprogrammed by various routes, but we didn't know really how the resulting cell types, how close they were to the actual target cell types. So a few years ago, we developed CellNet, which is a network biology platform that really uses bulk expression data to infer gene regulatory networks, and we use these to measure cell identity. We found that cells differentiated to target cell types, they looked to be developmentally immature. When we tried to take fully differentiated cells and convert them directly to another differentiated cell type, they also appeared developmentally immature, even though we were avoiding these developmental states. So this limits the practical utility of these engineered cells, and we have this critical knowledge gap, and that's how do we deconstruct cell identity to increase reprogramming efficiency and fidelity. So we're really looking at the fundamentals of cell identity in this talk. So the lineage reprogramming paradigm we work with is the conversion of mouse embryonic fibroblasts to induced or so-called induced hepatocytes. So this is a poster child for direct lineage reprogramming. So we knew that there was a mystery cell identity underlying these cells when we analyzed them via CellNet. And we went on to show that these cells actually have long-term functional liver and intestinal potential. So based on this and the self-renewal properties of these cells, we renamed them induced endoderm progenitors. And now this protocol is really useful because it forms a blueprint of cell identity. How do you take cells from one cell type to another fully differentiated cell type? But it's technically very challenging because so few cells successfully convert. So it was around about this time in 2015 that we adopted high throughput single cell sequencing technologies. But the issue was we were losing lineage relationship information in these experiments. So we developed the cell tagging approach that was so nicely introduced and delved into by Alon in the previous talk. So we developed this to really trace the origins of those successfully reprogrammed cells. And basically cell tagging is based on the introduction of random barcodes into cells. They're heritable, they're expressed within the transcriptome, so they're picked up in parallel and with the rest of the single cell transcriptome. And going through sequential rounds of cell tagging, periodic sequencing, we can build these lineage trees, so rather rudimentary lineage trees of the reprogramming process. But that's really taught us a lot more about the biology of this process. And through IEP reprogramming, we've defined these reprogrammed and dead-end trajectories that we've been digging into in a lot more detail. And now Alon and his group have defined so elegantly this concept of the state-fate relationship. And these lineage trees enable us to find these cells that are destined toward a particular reprogramming outcome across this process. So this is something I'm going to come back to at the end of the talk. So how are we going about extracting information from these early stages? But fundamentally, we think we need to approach this question thinking about the fundamental concept of cell identity. How is it established and how is it maintained, and what are the molecular drivers of this process? So we're coming back to classical developmental biology questions here. And we really come from the CellNet platform, which use bulk data to reconstruct or infer gene regulatory networks. So these are critical regulators of cell identity. So having knowledge of these networks across this process is essential. But also being able to achieve single cell resolution with this and break down that population heterogeneity is crucial. So the first component we have here, which is quite unrelated to the CellNet platform, is Capybara. So Capi, led by Wenjun, our graduate student who gave a poster yesterday, is designed to measure cell identity and fate transitions from single cell data. And the second platform is CellOracle. So this is gene regulatory network inference and in silico factor perturbation. And I'll focus on CellOracle for the rest of the talk. It's been led by Kenji, a postdoc in my lab, and assisted by Christy, a student. So both of these are up on GitHub and have been pre-printed, and we are set to update these preprints shortly. So the fundamentals of CellOracle. So we use a combination of single-cell RNA-seq and single-cell attack-seq to infer gene regulatory networks for each cluster that we identify across our reprogramming dataset, or differentiation or development, really whatever your interest is. And we can ask how GRNs change during these developmental or reprogramming processes, and we can look at how early network state is linked to eventual fate. Fundamentally, we can ask how transcription factors regulate cell identity across this process. So we can look for those transcription factors that position cells onto a specific trajectory from the very early reprogramming stages. So I completely agree with the data that Alon showed earlier, that these reprogramming trajectories are defined at an earlier stage. And we're going to take a deeper dive into that at the end of the talk. So how did we build CellOracle? So in the first step, we build a base GRN. So we take single cell attack-seq data. For this, we took the publicly available Kusanovich dataset, sorry. We use this to identify accessible promoter enhancer DNA sequences, and then we scan these regulatory elements for transcription factor binding motifs. And this creates a list of all potential regulatory connections between a TF and its target genes. So this represents the base gene regulatory network. So these are all the biologically plausible connections within a network. In the second step, we then use our single cell RNA-seq data to build a regularized machine learning model that predicts TF target gene relationships. So in this step, you would take your own single cell RNA-seq data. From the machine learning model fitting results, this presents the certainty of connection as a distribution, and we use this to remove the weak or inactive connections from the base GRN. So this creates your cell type or state-specific GRN or network configuration. So we've benchmarked CellOracle. So we've done this for hundreds of transcription factors across 5 different tissues, and we find that CellOracle, when we include the single cell attack-seq data to generate the base GRN, that it outperforms other approaches. And so we've got more details of this in our preprint and in our upcoming update of that preprint. So the first level of CellOracle output is where we leverage graph theory to provide an intuitive network analysis. So this can help you prioritize interesting transcription factors based on how they're connected within the network. So at this level, the output's very similar to scenic, which is an approach that we really respect in this lab. And it just helps you prioritize your factors, which you would then traditionally go and experimentally perturb. Now, the difference with CellOracle is that we can use our machine learning model that predicts TF target gene relationships to simulate the effects of transcription factor perturbation, either expression, overexpression, or knockout. And we then propagate that initial perturbation effect within the network to simulate the indirect effects of the original perturbation. And this produces a simulated expression matrix, which represents the future gene expression values following the transcription factor overexpression or knockout. And then we can project this direction of change in identity onto our dimensional reduction. So here, I'll go into this data in a little bit more detail later, but we're showing a predicted lineage switch during Zebrafish axial mesoderm formation. So just to give you some validation here, so we took this atlas of myeloid progenitor differentiation that was published in 2015. On the left, we've projected the wild type density here, differentiating from progenitors to erythrocytes and granulocytes and monocytes. On the right, we have CEBP alpha knockout that was included in the paper. And in the paper, they showed that CEBP alpha knockout blocks granulocyte and monocyte specification at these initial progenesis stages. So looking at the the CellOracle network analysis here, so we can see all the usual suspects in myeloid progenitor differentiation within the top 10 of these factors that have been ranked by betweenus centrality. So this is how well a transcription factor would propagate the signal through a network. We see CBP alpha in here. So just a little flavor of how you would use these network metrics. So moving on to the validation of the knockout phenotype. So we realized quite quickly that we needed to work with the forward momentum of differentiation in this case. So we calculate a gradient of pseudotime and project it onto the force atlas here. And we then calculate the inner product of this pseudotime gradient with the CellOracle CBP alpha knockout simulation. And this produces a cell density prediction in the CBP alpha knockout sample. So just to walk you through this, the areas in red are where we're predicting the perturbation effect to work with differentiation. So in these areas, we may expect that differentiation would be accelerated upon gene knockout. The areas in blue are where we're predicting the perturbation effect to work against the differentiation effect. So here we may expect differentiation to slow down, it could be blocked, or the cells could actually regress to an earlier state. Now, we don't claim to predict the exact forces in action here. It just gives you an idea of what the phenotype would be. We don't predict the precise gene expression as a result of the knockout. But when we compare this to the ground truth data, we see that in agreement, we predict this initial block at the level of progenitor, of the progenitor state. Those of you looking carefully here may see that we predict a later acceleration in differentiation, but we don't have the ground truth for this because the cells are blocked earlier in their differentiation. So this is something we never see. So this is something we take into account when we're interpreting these simulations. So we've been playing around recently with how to visualize these simulations, just so we can get some idea of what's happening with the simulated knockouts. So on the left here is the pseudotime, so a dynamic pseudotime of differentiation from progenitors into monocytes, granulocytes, and erythrocytes. And on the right, we have the GASA1 knockout simulation from CellOracle, and we see that erythrocyte differentiation is being blocked, as is granulocyte differentiation, which agrees with previous publications, which is another good sanity check for CellOracle. So just one more validation here, TAL1 knockout in hematopoiesis. So we use this fantastic single cell atlas of mouse gastrulation and early organogenesis. In this paper, knockout of TAL1 was also included. So TAL1 knockout embryos die at E9.5 from anemia. And it was shown from these single cell analyses that the TAL1 knockout cells within the chimeras do not contribute to hematopoiesis. So again, calculating our cell density prediction in the TAL1 knockout sample, CellOracle predicts accumulation of cells within the hemogenic endothelium. So this is where differentiation would be blocked, and also accumulation of cells in the allantois. And this is what we observe in the actual knockout single cell data sets. Okay, so I've shown you so far some very specific ground truth kind of validation, ground truth-based validation of CellOracle, but we really want to get into identifying new developmental regulators. So we've turned to this fantastic atlas of Zebrafish development from Jeff Farrell back in 2018. And we performed systematic knockout for all transcription factors with inferred connections to at least one other gene. And we've done this across Zebrafish development. So this results in about 230 transcription factors. So obviously there's a lot of developmental stages here, and we'll release a website on this soon so people can explore these data sets. But now we're specifically focusing on the axial mesoderm. So this is a really important patterning structure in the zebrafish embryo. We have specification of notochord and precaudal plate within the structure. So again, we calculate the pseudotime gradient across this, and focusing specifically on this notochord branch. We've ranked transcription factors according to degree centrality. So this is the number of connections they have to other genes. We see all the usual suspects in axial mesoderm development, and right at the top is the noto transcription factor. And so next, we again calculated the inner product of the gradient of pseudotime with the Noto knockout simulation to produce a cell density prediction. And CellOracle predicts this blocked notochord differentiation at early stages. Again, we get this later acceleration, but the cells would never get to that stage. Another way of looking at this, we can plot this inner product score against pseudotime, and you can see this early block, and I'll show you how we use this to rank factors shortly. Right now, the ground truth information for this, we're currently analyzing it. We just got data back from the floating head mutant, as it's called. But we know from previous work in the 90s that notochord differentiation is disrupted in the floating head mutant. So this is a Noto knockout, whereas precaudal plate develops normally here. So now how do we score novel regulators of axial mesoderm development? So as I said, plotting inner product score against pseudotime, we can look for this negative inner product score to signify that we have a block in development. So obviously, Noto knockout has a strong phenotype here. The androgen receptor does not have a strong predicted phenotype, just for comparison. So we can take the sum of the negative inner product score and then transform this to create a ranked list or a prioritized list of candidate notochord regulators. And here in red, we've got all the confirmed actors in notochord differentiation, which is a huge relief. But we've also found these novel factors that haven't been reported to play a role in notochord differentiation before. And so we're in the process of validating these, so knocking them out in axial mesoderm in collaboration with the Solnit-Kretzel lab. So, really a pioneer in zebrafish gastrulation. So finally, the last slide here is bringing it back to this concept of the state-fate relationship. So how are we approaching this problem? And I'm really excited to see CoSPAR. So I think we'll be visiting that very shortly. But as I said, with cell tagging, we have these two defined reprogramming trajectories, one to a reprogrammed state and one to a dead end state. And we can identify cells in the early stages of these trajectories. So these are the early states, and we can use CellOracle to link early network state to the eventual reprogramming outcome. And doing this, we found that from early stages, the networks are wired quite differently between these reprogrammed destined cells versus the dead end destined cells. And here we are really focusing on these lineage restricted cells and not looking at the cells that transition to both lineages. We're also not looking at the cells that completely fail to reprogram, so they remain as fibroblasts. So that's a question for another day that we'll dig into. But we found on the reprogrammed destined cells, so at these early stages, that FOS, transcription factor that's really involved in so many biological processes, was highly connected on the reprogrammed trajectory, so in these reprogrammed destined cells from early stages. And so from CellOracle simulations, we simulated overexpression of FOS, so adding that to the FOXA1 HNF4 alpha reprogramming cocktail. And we found that CellOracle predicted that cells would move from early transition phases to transition phases, but also move from dead-end states into a reprogrammed state and increase the yield of successfully reprogrammed cells. And we've been able to validate this experimentally, looking at colony formation assays, and also digging a little bit more into the mechanism. So looking at the inferred targets of FOS, we've been able to find a role for YAP and hippo signaling in this process. So this just gives you a little flavor of how we can use this network biology and integrate it into these lineage tracing approaches. And with that, I'd like to thank the lab, just a really great creative group of people to work with. I'd also like to thank the community as well, because this is such a fun community to work with. And I hope today you've seen how we're all working with the same data sets to approach these problems from different directions and really supporting each other, which is, incredibly exciting to me. Our cell tagging resources are all available. CellOracle and CAPI are up on GitHub. And thank you to our funders. And we're also looking for postdocs. So please get in touch and I'll be happy to take any questions.
Thank you, Samantha, for a really, really wonderful talk. A number of questions, and I'm just going to start by asking one that I have. So so it's really cool to be able to identify candidates. And I guess one of the things I'm wondering is uh, of the candidates. So, I mean, there's of course the question of validation and how many of the candidates are, true or not or whatever. But I guess one of the questions that I have is, if you just take the list at face value, can you say something about what the differences are between things that are like barriers to a cell fate transition and things that are drivers of the cell fate transition? Um, can you distinguish them? And if you can distinguish them, then, are there any systematic differences between them?
Yeah, that's a really good question. So in the reprogramming we found on the dead end, ZEB1 is highly connected. So this, our hypothesis is that's keeping cells in a mesenchymal state, whereas to be successfully reprogrammed, they need to flip over into this epithelial state. So that's actively preventing them reprogramming. But then when you're talking about removing barriers, fundamentally we're using attack-seq data to build this base GRN. So we're looking at more of from an active perspective there, looking at open chromatin.
Right.
Can we, and this is the nice thing about CellOracle, with that base GRN, you can build other kind of modalities into that. So we can start looking more at repression. And this is a question that comes up a lot, actually.
Yeah. I guess maybe a sort of related question is, from Carlo Leonardi, is, does CellOracle take into consideration the false positive rate of single cell attacks?
It's a good question. The false positive rate. So we're using Cicero to construct the GRN. That's something I'll have to think about in more detail. Definitely. Yeah. Aside from single cell attack-seq, you can use bulk attack-seq. You can also dispense with attack-seq altogether. So just looking at promoted databases, because obviously with the Zebrafish development, we don't have any attack-seq for that yet. We don't have a complete atlas for that. So we've really just used a UCSC promoted database to build the base GRN from that. So, one thing we can do is like really compare these different methods, because we're now generating the single cell attack-seq data for that and look at the performance there. But looking at the accuracy of CellOracle right now, it's doing fairly well. But can we improve on that?
Joshua Thyssen asks, can CellOracle make predictions in situations where knockout of a signaling molecule would affect expression function of more than one transcription factor?
Oh, that is a good question. Yeah, because obviously right now we're completely transcription factor centric, which is somewhat of a limitation. So if you have some kind of signal perturbation, it affects the expression of several transcription factors. If you know what those values are, you should be able to simulate that. So we've been playing around with doing double, triple knockouts, combining overexpression simulations with knockout simulations. Kenji does not like us doing that, so he's very conservative with the use of CellOracle. So he urges caution with that, but we've had a lot of fun with it, and we're quite optimistic actually. So that's something we'd like to explore more. And on the subject earlier of showing when these approaches fail, I think that's a really good point. So we are, performing
my de facto question.
Yeah. So performing these systematic TF knockout, so particularly when in hematopoiesis, we're finding when CellOracle cannot make accurate predictions, or where there's just no prediction at all. And it's more often than not when we don't have any TF binding information. So it's just not reflected in that base GRN, and that's where CellOracle fails. So
cool. Katie Galloway asks, it's a fantastic talk. It's absolutely true. Given that FOS is an immediate early gene, do other immediate early genes or MAPK-induced genes have the same predictions for promoting reprogramming as FOS?
So we see the same with June as well. So that immediately started ringing alarm bells with me, thinking, this is just some kind of stress response from dissociating the cells. So, Kenji's been really clear to show, even without cell dissociation, that we see these immediate early genes kind of fire up at the beginning of reprogramming. And we think from the simulations, they might have a slightly different role early in reprogramming than they do in the middle of reprogramming. So that's something we're looking into in more detail now.
Cool. We'll just do one quick question before we break, and this is from Amanda Haupt. Very cool. Can this predict outcomes of multiple manipulations, like knocking down one thing and activating another?
Yes. So this is we don't have the validations there right now, but from other projects in the lab, when people have have their data and they're trying to kind of figure out more of the mechanism, they're doing that. So knocking out one gene, overexpressing another, and intuitively it's making sense, but always do the experimental validation.
Awesome. Yes, wise words.